yeah, thanks so much for having me. I've put together a talk which is a little different for me. I'm going to certainly talk about some things in cell biology and biomedical science, but I wanted to kind of take it back a bit and get really into the basics of analyzing images and working with images, and specifically, how can we use this in biological sciences applications. But hopefully by the end of my talk, you will kind of see that anytime you have images, that they are a rich source of information and that you can do a lot of stuff and extract a lot of information out of them. So I was just telling Torsten that people in Bath are also chemists are using automated image analysis to look at the crystallization and solubility of small molecules. So there's a lot of things that you can do on every scale. So I really wanted to kind of get into some of the basics of just computer vision image processing, because these will be terms that you might come across in a variety of different contexts, even if you don't end up doing cell microscopy. So I specialize in microscopy. So I'm a cell biologist, which means my interest is really looking at life on the scale of a cell. So I also just love to look at cells in microscopes. So microscope images are extremely information rich. So what we're seeing here is an array representing one field of view from each well of a 384 well plate that was seeded with a panel of 22 different breast cancer cell lines and imaged using a high content microscope. And if we zoom in on some of the wells, you can see, oh, sorry, this is pictures, they're very pixelated, but that there are quite a lot of diversity amongst the cells. Actually, this one kind of looks better. Let's just look at this one for now. So there's actually quite a lot of diversity. So cells are at different densities, so they're growing at different rates. They have different shapes. The proteins that we have labeled here are localized in different ways. And if we zoom in on that a little bit, you can kind of see that some of the cells are more green and some of them are more blue. That's because of the different stains that we've applied. So this one data set is just using one multi-well plate, so has 384 wells, has 22 cell lines, and each of those has 2 treatment conditions represented per cell line. So in all, just this one plate has about 12,000 fields of view in 3 fluorescent label channels. So this is an absolutely vast number of images already. And in each field of view, there's anywhere from maybe a few or 10 to hundreds of cells per field. So this is absolutely, when we get down to the level of looking at individuals, this is a massive amount of cells. And now we have then gone back and used the processes that I'm going to talk to you about in a minute to extract lots of information about the individual cells. So we look at things like their morphology, their shape, we look at their context, how many neighbors do they have around, and then we look at all of the things, obviously, that we have stained using molecular dyes or immunofluorescence. So what we get out of this are just absolutely massive text files with anywhere from thousands to hundreds of thousands to millions of rows, and anywhere from dozens to hundreds of columns. So what do we do with this information? Well, this data set, excuse me, I used in a paper a few years ago to profile breast cancer cell lines based on their morphology. So what we found was that cells that had similar genotypes actually clustered together based on features of their morphology. And we identified distinct subtypes within these different genetic classes from cells that were derived from the inner layer, the luminal layer of the breast milk duct, and cells which were derived from the outer layer, the myoepithelial layer. So we were able to actually recapitulate the genetic classes and also identify some differences within classes as well. So here in these luminal classes, we found some cells had this very kind of flat spread morphology, and other cells were these very little kind of round grape-like clusters of cells. So then we used a combination of image analysis and other methods that I don't have time to go into, but have linked the paper. to show that not only could we see these genetic clusters and we could see these morphological types within classes, but that cells in the different morphological classes would respond differently to inflammatory cytokines, and they would do this in a predictable way. And then we could actually manipulate the morphology of the cells and see some predicted changes in how they would respond to inflammatory cytokines. So this is all very interesting, but how did we actually go about doing this? So we did this using automated cell detection and automated measurements. So just in brief, we started with identifying cell nuclei and then identifying the bodies of the cells. You can also use other markers to label organelles, such as like the mitochondria or the ER or the Golgi. And then from these stencils, these regions that we've defined, then we can measure all sorts of things. So we can measure the intensity, we can measure the texture, sort of how are the pixels, how are the stains distributed. We can measure the context, so how are the cells touching other cells? Are they clumped together in colonies? Are they spread out? You know, what is their relative position? And so we end up with measurements where we've got individual cells and anywhere from, you know, hundreds of features per cell. So now let's talk about how this is possible. And I really want to take a step back at first. Oh, we also applied some filters, some quality control filters to get rid of cells which were dead or missegmented. So let me take a step back and ask ourselves, what do we actually mean when we talk about images? So what actually is an image? So an image is an array of values. A 2D image is an XY array. 3D stack of images would be XYZ, XYZ. This is all it is. An image is just a list of numbers with some information about how they're organized in X and Y. And this seems, I don't know, this seems sort of basic, but actually when I really realized the sort of implications and ramifications of this, it was very sort of like, oh, galaxy brain, right? Because any array of numbers could be an image, and an image could be made of any kind of an array of numbers. It's not like just that you've taken a picture with a camera. Like what the computer sees is different from what our brains see. So it sees pixels in an array. So each pixel is one point, which has a position in the array and a value, some sort of numerical value. So that could be an 8-bit, a 12-bit, a 16-bit, or even a 34-bit value. So an 8-bit would be a value from 0 to 255, right? Because there's 256 possible values. So we can also have a color picture where you've got, say, red, green, and blue, or really any number of color channels. And this is just where this picture is taken from. So a computer screen, an image that you see on a computer screen, that's almost always going to show you an 8-bit depth if you're looking at a grayscale image. So the human eye can see something like 30 to 100 shades of gray reliably, and this is going to depend on things like the brightness and the contrast and, you know, your own personal human brain. So that means that the computer, especially if it's looking at a 12-bit or a 16-bit depth image, which is what we would tend to get from a microscope or an MRI, that means that the computer can see a lot more than we can actually perceive when we are shown an image. That said, human beings are much better at finding things in images, especially when we're working with incomplete information. And we're much better at doing this than computers. Although, as we'll get to at the end of the talk, with advances in machine learning, that is changing pretty quickly. So I just want to have a little think about how do we find things in images? And so this is a concept called in computer vision and image processing is segmentation. So how do we divide an image like this, which is just blah, as far as the computer is concerned, a big array of numbers. How do we segment it into cars and streetlights and people and the sky? How do we detect and define objects in an image? So I sometimes think like, how do I do that? You know, we can do it with an artificial neural network. Well, how does my organic neural network actually do that? So we look at things like shapes, we look at things like edges, we look at color, we look at texture, we look at shading. We have a whole set of priors that we know about the world, and we have a set of kind of archetypes and patterns. We know what a car looks like because we have seen lots of different kinds of cars. So when we see a new car, even if it's rotated, even if it's blue or green, we're seeing some qualities about it, and then our brains are going beep boop 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 and matching those patterns to things that we already know that we define as a car. So segmentation is everything. Once you have good segmentation, you can measure anything and you can measure everything. So from this image, once we have defined that this is a car, this is a thing, this is a thing, this is a thing, and oh, okay, like this thing is a car, this thing is also a car, this blob is a human, this blob is a human. Now we can measure things about cars, we can measure things about humans, we can count the cars, we can count the humans, etc., etc. So segmentation is really the basis of all of this going forward. So how do we do segmentation in, say, microscopy images? Well, a straightforward way to do it is use your organic neural network and just manually draw regions of interest around objects. But as you can imagine, this is not super efficient. I can tell you my PhD involved a lot of this because it was a while ago. I was looking for things which were difficult to get the computer software that was available at the time to reliably identify. So I relied on my organic neural network and I did a lot of drawing regions of interest. I could now, if I was to do all of that again, it would take me a lot less time. I'd have a lot more data. So let's steer clear of manually drawing regions of interest around objects. Let's see if we can automate the process. So the first kind of automation that we can do if we're looking for objects which are bright on a dark background, or could be objects which are dark on a bright background, is to apply some sort of pixel intensity cutoff. So if you look at this image, these are some cell nuclei stained with a label that labels DNA. And what I've done here is just in ImageJ or Fiji, I've just opened the threshold module, and I've moved the threshold around. So I said, okay, any pixel that is brighter than this line here, I think it's set at 117, color that in red. And if we look at this line, if we make a profile of that line, then we can see that as we move across that line, it's very dark. Oh, suddenly very bright as we encounter this object. Another bright spot where we encounter another object, but which is dimmer than the first, and again a dark spot, and then another bright spot. So you can also think of an image with pixels that have intensity values. You can think of it as a landscape, like a topographical landscape. And here we've just taken like that would be like an XZ slice. So we're looking at now, instead of dark spots and bright spots, we're looking at low points and high points. And what I've done here with this threshold is say, okay, any hills that are higher than whatever it was, 117, call those a nucleus. But obviously, what am I doing? I'm missing a lot of nuclei here. I have made this threshold too high, so I can go back, make this threshold lower, and now I'm grabbing pretty much all of the nuclei here. Also, I'm grabbing some schmutz in the background. But that's basically thresholds. Typically your first point of call when you're looking for foreground versus background. So there are some more sophisticated ways of doing this than just looking at this topographical map and drawing some lines through it. So one of these is called Otsu's method, after a person called Otsu. And this is saying, okay, look at this image, and it's got pixels of varying intensities. Find a way to assign pixels to classes. In this case would be like, object class versus background class, foreground class versus background class. So say this is the foreground object, this is the background. So find a way to assign pixels to classes so that the variance between the two classes is greater than the variance within the class. So in a binary image, this is super easy. So if we look at a histogram of, this would be the frequency, this would be the pixel intensity, right? There's only 2 peaks. So you can draw your line anywhere in here. It's going to work. This image is a little bit noisy. It's not binary, but there's still pretty good cutoffs. So the pixels in the object have a certain Gaussian, and the pixels in the background have a certain Gaussian. And so Otsu's method would say, great, okay, cool, very easy. Draw the line here. This becomes a problem, though, when we get to very noisy images. So here, the noise, the variation in the pixels in the foreground and the variation in the pixels in the background are quite similar. So when we look at the histogram, this is going to be hard to segment if we just are trying to draw some sort of threshold line. So Otsu assumes that there are 2 classes, but you can also tell it to define like n number of classes. Just makes the maths a little bit harder. So what can we do here to get this method to work? I'm not going to go into the ins and outs of the math for this, but there is, if you want to check that out, this Coursera course section on Otsu is really nice for it. It goes through all of the maths. So what we can do is we can take this noisy image and we can do some smoothing. So say we can apply a Gaussian filter. And what we're doing here is kind of taking a sliding window, so some sort of kernel, and we're just taking an average. It's like the airbrush tool, right? So you've probably all used Photoshop. You take your airbrush tool and like, oh, now we've removed all the spots from our face. Once we've done that, if we look at the histogram now, we can find a nice place to draw the line and segment. Because it's giving us a much cleaner bimodal distribution of pixel intensities. And so then we can draw that line and define foreground versus background. So Otsu's method doesn't take into the relationship in space of where the pixels are with one another. It's just doing that kind of drawing a line, but it's doing it using the histogram rather than the kind of topographical feature plot. So Otsu's will fail if the illumination is really uneven. So here we have a picture of some handwriting, and you can see that it's quite bright in the middle and dim out to the side. And if you've ever worked with a fluorescence microscope, you'll be very familiar with this uneven illumination, because just the vagaries of physics, the field of view is always much brighter in the center than it is out to the sides. So if you try a global Otsu with this, is you're going to get lovely segmentation of the handwriting in the middle, and then it's just going to say, oh, everything out here is just black. So there are some things that we can do. So uneven illumination can be corrected using a pre-processing step, where we can either like do some background subtraction, or we can like fit a polynomial. We can get a mathematical function that describes a landscape with a hill in the middle and some dark patches out to the side, and then divide by that, and that's going to even up and correct the illumination. So Otsu's then will now work really nicely. We can also overcome, to a certain extent, local differences using a sliding window. So instead of doing the Otsu line drawing for the whole image, we can take a sliding window of whatever size we want. So it's called the adaptive window, and we can just do the Otsu here, slide it along, and then that's going to give us a much better result, because now these global changes in the illumination aren't going to really affect where that line is drawn between the background and the foreground. There's another method that if you use a program called Cell Profiler, which is something that we use in my lab a lot. It's a freeware that is very commonly used for cell biology. You will also see something called the minimum cross entropy segmentation. This is very similar to Otsu, because it's looking for some sort of line to draw a threshold to classify pixels as object or background. But instead of kind of looking at the variance, the within class and between class variance, it uses probability distributions. This gets very mathsy, which we don't have time to go into, and I'll probably embarrass myself if I try to describe. With minimum cross entropy, though, you can also use the sliding window if you've got uneven illumination. So these are good tricks. These are good tricks of the trade for working with microscopy images, but also, these are the same processes that are used in photography, that are used in cinema to, like, identify Spider-Man against a background, and then say, oh, let's take Spider-Man, and oh, he's in New York, and now let's put Spider-Man in outer space. So all of these methods are really widely used. Okay, so now we've got, each pixel has now been assigned to a class. Say we've done this nicely. So we've got our nuclei here. We've got nuclei in red, background in black. And here we have a cell body stain. And so we've got figured out a threshold to draw so that we've got cell bodies versus background. So this is, we've now segmented our image because each pixel in this image knows what class it belongs to. Is it a background class? Is it a nucleus class? Is it a cell class, which has some overlap, obviously, with the nucleus class? So this is what's called semantic segmentation. So it's a kind of, each pixel has an identity. But what we don't have here are any sort of object identities. So like a pixel here doesn't know if it's in a group with these pixels or with all of these other pixels around here. So for that, we want something called instance segmentation. And that's where groups of pixels that are in the same class are grouped into discrete objects. So here you can see in every different color outline is one nucleus, and different color outlines are representing one cell. Okay, so this is where it gets a little more tricky. The threshold thing, the semantic segmentation is kind of the easy part. This is where it gets to be a little more tricky. So one of the kind of most popular, oldest ways of doing this is what's called watershed algorithm segmentation. So picture the topological plot that we were talking about before, where like the nuclei were these mesas or these hills, and the background were the valley. Okay, so now flip that up on its head so that the nuclei are now the valleys and the background is the land. And now let's imagine some raindrops falling on that topographical landscape. So what's going to happen is that the water is going to pool in the valleys. Or they're called catchment basins in geography. So as the water rises, right, as we pour more rain, the water will rise in these little pools. So here, this little local minima, which used to be our local maximum of where a nucleus is. So as the water rises, the pools begin to touch. But this algorithm is going to draw lines between them so that it's defining this region, say, as cell number 1, this region is cell number 2, this is cell number 3, even when they touch. So in order to do this, watershed algorithm uses something called a distance transformation. And this is, I don't know, it's also like galaxy brain to me, because you can transform an array of pixels into a different array of pixels. And probably if you are a person who's really conversant in like matrix mathematics, this is like not mind-blowing and exciting for you, but I am not that person, so I find it quite exciting. So in the distance transformation, we're taking the image, the array of pixels, which now has two classes, say foreground and background class, and then we're going to transform the value of each little spot on this array into something else. And that something else here is going to be the distance to the nearest background pixel. So for the ones on the outer side here, well, it's 0 because it's 1, sorry, because they are touching background pixels, so they have a distance of 1. The ones in the middle here, this has to travel now 2. So this pixel would be transformed from a value of 1 into a value of 2. And this one has to travel 3 in order to hit a background, so that becomes a value of 3. And so now we have here, if we had, this started out as a silhouette of a horse, now we have this sort of like horse skeleton, which is the distance transformation. So this is the way that the watershed algorithm is going to decide kind of where to draw these lines when it's drawing the lines between the catchment basins. So start with a binary image, do the distance transformation, and then from this distance transformation, we get these watershed lines. So the lines that are drawn between the objects, and we can see this is like, here's the 3D topographical map of the original image. And now we will get segmentation based on the combination of this binary image and the watershed. So here's just an example of this original image of some nuclei, the binary mask, the distance mask, and then now here is what's called the vector image, the vector mask, where each of these objects has now got its own identity. And so if we were to look at what does a computer see when it looks at this image, imagine that it's grayscale, it would see a lot of pixels which are black. You know, if this is object, call it object number 1, these pixels would be a 1, these pixels would be a 2, object number 3, these pixels would be a 3, etc. So this is how we actually, one of the ways that we can get this instance segmentation. So watershed algorithm, these values are defined by the local maxima. So here is just me using ImageJ to find local maxima in the original image of some nuclei. So this is just like showing me where are the peaks, and you can ask it to, you know, give you different numbers of peaks, different cutoffs of peaks. So it is sensitive to how many and how far apart these peaks can be. So you can decide to like suppress them or merge them or average them in order to get the right segmentation. Because if you don't, you can end up with objects that are merged together where they shouldn't be, or objects that are split in half, because like this one, it happens to have 2 bright spots that are in the same object. So there are some other ways that we can kind of boost our segmentation, boost our object detection. So one is looking for edges. So there are various edge detection filters, and what that looks for are kind of large jumps in value between pixels, kind of sudden differences in elevation between pixels or kernels. So you can say, okay, look not just from, you know, 1 pixel to the pixel next door, but look at groups of 3 or groups of 5 or groups of 10. So this is really useful for, like, say, nuclei, for example, where there's a nice high signal to noise ratio, there's a bright stain on a dark background, and they've got really clear borders. It doesn't work quite as well for something like this cytoplasmic stain, which it has gradients, a lot of gradients, rather than these kind of discrete borders. So this is an edge detection filter, and you see it just looks kind of like wavy spaghetti. Yeah, you might be able to use it, but it's not going to necessarily give you as much help as the other one. Okay, so just in summary, cell segmentation, or any kind of segmentation. So we want to assign pixels to classes, say foreground versus background, define thresholds, and we can do that globally or we can do that locally. And then we want to group our foreground pixels into defined objects. And we can do this with distance transformation, watershed, looking for edges, and obviously combining these different methods into like kind of more sophisticated algorithms. Okay, this is very great, and this works a lot of the time, but it doesn't always work. So sometimes images are hard to segment, and I'm going to give you some examples from my life, which are unexpectedly shaped nuclei. So these nuclei are from cells which have been transfected with a small interfering RNA that targets lamin A, a nuclear structural protein. So they are very weird shapes. Some of them are these kind of normal jelly beans, but we've got ones that are shaped like a C. We've got ones with a big hole punched in the middle. We've got ones which are like, got a big sort of line through them. Trying to segment these using the kind of watershed is a nightmare because, like say this one's going to have local maxima on either side, and then when we do the watershed, it's going to be like, oh, no, we should definitely draw a line there. So it's always going to missegment these cells. This is really difficult. Here's another example where I've got some cells which have they are expressing a fluorescently tagged nuclear protein, but because it's only expressed, it's the endogenous protein, so there's not that much of the stain. There's no signal amplification on the fluorescent signal here. And because they're live cells, which I was imaging over time, we couldn't take a very long exposure because then the cells will suffer phototoxicity because you're just, zapping them with a lot of light, you're exciting fluorophores, you've got electrons hither and yon, you've got reactive oxygen species being generated. Cells get very unhappy when you do this to them. So how on earth do we get the computer to identify this as one nucleus and to identify, say, this is a nucleus where we've got a combination of uneven illumination and really low signal to noise. So here's where we use some deep learning strategies. And this is stuff that's really come a long way in the last decade. And if I'd had it when I was a PhD student, I would have probably graduated sooner, because my life would just been a lot more streamlined. So here in the deep learning scenarios, we're going to have a human being tell the software what it is that they're looking for. And then we're going to ask the software to just like make it so. Just figure out a way to classify pixels and objects based on what I have told you, based on the training. So here's one software that I use. It's free. You can just download it. It's called Elastic, with a weird spelling. The documentation is actually really good for Elastic, so you can. It's quite a small learning curve, I think. So what you do is you import your images, and then you just make some scribbles. And so I've said, okay, define some backgrounds, define some nuclei. So background in red, nuclei, draw some blue lines, and then you hit live update, and it gives you a prediction of what it thinks that nuclei should be, for example. So here we're looking at the result of its segmentation. So in yellow are its predictions of what nuclei are, but I've also got this uncertainty tag on. So it's also telling me, these pixels that are in cyan are pixels that I am not certain about. Like maybe they're part of a nucleus, maybe they're part of the background, I'm not sure. So, but this is all very iterative. So I can just keep training it. I can just keep feeding it more information until it hits some plateau where it's not going to get any better. So in this case, what I've done is I've said, okay, you know what, like draw a little line here because that's background. These are two objects, they shouldn't be touching. Live update and see that the situation improves quite significantly. So by annotating images, what you do is you train the neural network, which is running under the hood. You don't need to know anything about it, but you train it by annotation until you're satisfied. And when you're looking at like a big data set, you want to kind of have a subset of images that you train on, make sure that they're kind of representative of everything that you're seeing as a whole, because otherwise, you might train on images from the left-hand side of the plate, but the right-hand side of the plate had a little bit more or less staining, and so the segmentation might not be optimal. So when you're doing this kind of training, you want to make sure that you're kind of training it on a training data set that is representative of the larger data set. And so there are some quality control steps that go into this as well. So here is an example of, this is something I tried to do with some phase contrast, some bright field movies. So these are some movies that I took of cells just using phase contrast microscopy. These are notoriously difficult to segment, right? Because instead of looking at bright spots on a dark background, high signal to noise, easy to draw a threshold, we're looking at, splotchy shades of gray on a splotchy shades of gray background, right? It's like the monstrous noise monster. But I used this deep learning web browser interface. Unfortunately, this was one that you have to pay for, but I trained it to try and identify cells which were undergoing mitosis. So I can play the movie. So here's the original image. And then when white spots pop up here, that is where the software think that cells are dividing. It was trained using only static images. So it's kind of over predicting things. It's thinking that some things are mitoses even when it might be a cell that's just on the move, and so it's rounding up and moving away, and occasionally a cell dies and it thinks that that's a mitosis as well. So project that hopefully actually a graduate student is going to be working on is doing this deep learning also in the time dimension, which is quite similar to doing deep learning for segmenting 3D Z stacks. But I think maybe there's going to be some, it all sounds very easy, but it probably won't actually be as easy in practice. But all things considered, it's not bad. It's definitely work withable. And so this is major progress over a human being sitting and trying to spot every single cell division, because as you see towards the end of the movie, there's just hundreds and hundreds and hundreds of them. So deep learning is also used for classifying not just pixels, but also objects. So here is an example that's from Zeiss's Appear interface, which this is also free to use. You just have to register. And what you can do is do your own image annotations, and then you submit the job. Downside is elastic, you install it on your computer, you use it in real time. This one, it's all so cloud-based, so you submit a job and it comes back to you the next morning. But here we are looking at a transmission electron micrograph, and the annotator is defining lipid droplets and mitochondria and lysosomes by doing this annotation. And then the neural network that's running under the hood is going to come up with a way to predict, okay, that this is a mitochondrion, this is a mitochondrion, maybe this is a lysosome, well, this is another lysosome, maybe this is a lipid droplet. And kind of using this black box of stuff that's happening all in the background in order to do it, rather than the human being trying to come up with some sort of set of thresholds or features or parameters or bins that describe these different objects. Okay, so just to sum up this bit, we can also use deep learning for basic pixel classification. So when defining thresholds, either globally or locally, is failing us, then deep learning can ride to the rescue. And it's also really good for classifying objects into types, especially when it's not, like, it might be quite obvious to your human brain, but it's not obvious numerically why this is a mitochondrion and this is a lipid droplet or a lysosome. I mean, I can tell, it's kind of looks stripy, but this one isn't the same stripiness as this one. But there is something. Obviously, there's something, because the human annotator can recognize those patterns. But just because we recognize a pattern doesn't mean that we know what it is that we're seeing, which I think is also quite an interesting kind of philosophical construct that you can think about when you're coming home from the pub, maybe. So let me just give you an example again from real life of using this approach to derive cell cycle information from microscopy images. And this is something that some students in my group, undergraduate students, have been doing. So it might seem very sophisticated, but I've had undergrads doing it within a couple of months. So when we look at this nucleus stain image, so what we've done is we've labeled the DNA with a fluorescent molecule. So we can use that to find where the nuclei are, but we can also measure the intensity. And we can measure all sorts of things about the intensity. We can measure the average intensity, the average value of pixels in that region. We can measure the sum of all pixels in that region. And that's something called the integrated intensity, the total intensity. And that total or integrated intensity number gives us a rough and ready readout of the amount of DNA, right? Because each molecule of dye binds to some amount of DNA and gives off photons when we excite it with fluorescent light, which are gathered by the detector. And then that gives us, the number of photons is proportional to the number of molecules proportional to the number of DNA molecules. So if we plot a histogram of the total nuclear DNA stain intensity, and this is just a frequency histogram, we can see that there are two peaks, and those peaks correspond to cells in the G1 and the G2 phases of the cell cycle. So remember, the cell cycle, cells start off with one copy of the genome, grow, hit a point where they decide to replicate their DNA, so that's called the synthesis or S phase. Then they come to the end of that, do a little bit more growing, hanging out, maybe repairing any DNA damage that happened during synthesis. And when they're in this stage, they've got two copies of the genome because they have synthesized all the DNA, but they haven't yet replicated, they haven't yet divided into two cells, which happens in mitosis. So we have two peaks. We've got the G1 peak, we've got the G2 peak, and then we've got this kind of interim kind of plateau, which are the cells that have more than one copy but less than 2 copies of the genome. So these cells are in S phase. But again, this is rough and ready. There are, it's sensitive to missegmentation. Cells might be polyploid. They might have failed the cytokinesis, the division stage at some point, and ended up with 2 copies of the genome. So it's pretty good. It's not perfect. So we want to make it a little better by looking at some other stains. So here we are looking at pCNA, which stands for proliferating cell nuclear antigen. It's part of the DNA replication complex. And when the cells are in S phase, when they're replicating their DNA, the pattern of the pCNA goes from being kind of more homogeneous and diffuse within the nucleus to being very punctate and spotty. Cells which are like not even thinking about dividing are not even expressing pCNA. So we can also distinguish cells which are not even in the cell cycle at all, called a G0, so like a quiescent cell or a senescent cell. And then we've also got another stain, which is cyclin A. So cyclin A begins to accumulate at the very start of S phase and gets brighter and brighter and brighter and brighter and brighter as the cells go along until mitosis. So with this combination of stains, we can do an overlay. So here we've got the DNA in blue, the pCNA in green, and the cyclin A in red. And I do apologize to anybody who might be colorblind. So we can use these overlays to classify cells. So if something has basically only the blue stain, nothing else, it's G0. If it's blue and a little bit green, but the green is smooth, we'll call that G1. When there's green and it's spotty, that's when the cells are in S phase, if it's got that speckly texture. Depending on how much red stain there is how far along in S phase they are. And when we get to cells which are in G2, they are both blue and green and red, but not spotty. And then we've got cells, I don't know if you can see this, which are in mitosis and either in an early stage of mitosis before the DNA has gone from the center of the cell into 2 daughters, or at a later stage of mitosis. So the human, in this case myself or a student, can then annotate a bunch of images. So using the deep learning to identify the objects and then also to assign them classes, to annotate a class. So saying this is a G2, this is a mitotic, this is a late S phase. And then we have actually come up with quite a nice representation and classification of the cells into these different cell cycle stages. And here's just an example of some of the data that one of my students got out here. So we were looking at, so remember the lamin A depleted cells, which had the very wonky misshapen nuclei. Well, it turns out that not only are the nuclei weird shaped, but they also have some altered growth kinetics. And using this method, the student was able to find that there were significant differences in the proportion of cells which were in the different cell cycle stages when the cells were plated either at a low density or high density with or without lamin A. So that's just a nice little proof of concept, but we've actually got 1, 2, 3, 4, 5, 6, 7, and we could even push that, I think, to 8. I think we'd have another mitotic class. So just using 3 stains, now we've got 8 different classes of cells that we can compare between conditions. Okay, so segmentation and classification by deep learning. So I've talked about the semantic segmentation and the instance segmentation. Deep learning can be used obviously for all of those things, combinations of those things, and it can also be used to just compare one image to another and classify an image as this is a picture of a cat. And now I could group it with all other pictures of cats. And we're not worried about like where the cat is, we just know that it's a cat picture. We could also say, it's a picture of a cat, here the cat is. Or we could say, there's a picture, it's got a cat in it and also 2 dogs. So there's a lot that we can do with deep learning. So let me just give you, so I can move my own thing here, some examples of this. So one thing that this type of deep learning is become very popular for histology sections. So doing things in histology sections like identifying blood vessels or identifying tumor cells or stroma or fat droplets or lesions. These are pictures that are taken using a software called QPath, which is also a freebie, which is pretty cool. It's also running deep learning under the hood, and it's a user annotated type of system where the user would say, okay, this is a whatever it is, a kidney tubule, what have you. And these dark spots are cells which are positive for a proliferation marker, or here is a complex tissue and there are different things in amongst it. And now what we want to do is maybe we want to compare, well, how many cells are in this bit of tissue, how many cells are in this bit of tissue, which ones are labeled with a certain label, what's the area, say it's a tumor section, what's the area of the tumor. Is that tumor infiltrated by immune cells, for example? So it's a really powerful way of doing that. And it's specifically built for histology sections. So it's made to handle very large images that have a lot of information and usually several colors in them. So that's an example that's really, it's using both the semantic and the instance segmentation. But don't even necessarily have to do that on microscopy samples. So this is also, deep learning's very popular in, getting very popular for medical imaging. So identifying things, say in a CT or an MRI scan. So, here, this is, I think, looking at like a transverse section of a human body, and it's identifying different organs and different structures. Here we're looking at brains and looking for areas of brain injury. So identifying, tumors or lesions or areas of hemorrhage. And AI for medical imaging and diagnostics is a rapidly growing business. So the Cellular program that I talked to you about before, I had kind of a free trial with them, but their market is really, people looking at medical images. These images tend to be quite big. They are often, shades of gray, where, an expert radiologist, if you've ever been, and had any sort of MRI or a scan, you look at it and it just looks like a blob, right? You need many years of training generally to find things and interpret these images. So AI is really getting to be, is really a growth industry for medical imaging. So we can look also, so this is instance segmentation, but deep learning is also being used in this kind of just classification without actually, drilling down into like, what are we seeing? So here's an example of looking at tumor versus non-tumor tissue histology sections, and it was trained on, some ground truth of samples that we know are tumors and samples that we know were not tumors, and applying a classification just to the image. Like, we don't necessarily even know what's in the image, but we're just predicting that, these ones are not tumors and others are tumors. And that's obviously using a training set, using a ground truth, and then bringing in unknowns and asking, okay, do they fit the class that's defined as the tumors or as the non-tumors? And here's another example of looking actually just at photographs of moles and trying to decide, are they, bad, scary moles, or are they moles that you can ignore and get on with your life? Because again, it takes really a pretty expert eye to look at a mole and know if it's a melanoma or a squamous cell or just a weird, just like a little weird mole. So really powerful potential to aid in diagnosis and prognosis. But I wonder, like, can we replace the human expertise? Because somebody needs to train these. And when we are working with AI that has been trained by human users, I think it's really important, especially in the medical context, to ask ourselves, what kind of unknown biases have crept in when we're doing the training? maybe there are certain things that exist in maybe the data set, the training data set, does not accurately represent all of the, sections, the photos or the sections that we're going to see in the world. Maybe the human observers, were pretty good at their job, but consistently missed something. Maybe if all of these dermal photographs were taken from people with light skin, are they going to miss melanoma in a dark-skinned person? So AI is a very useful tool, but like any tool, it is wielded by humans, and humans are fallible entities. So it's always good to be skeptical and think about that. So I don't want to go into this too much, but I want to talk a little bit about these neural networks, like what's going on under the hood. So the most common of these is called a convolutional neural networks, or CNNs. So they're very good at classification, they're very efficient. So it takes an input, it does a bunch of convolutions to that image, and gives you an output. What the heck is going on here? Okay, so what it does is it takes the original image, and then it puts that image through various filters. So remember those edge detection images that I showed you? So that was using an edge detection filter. And these various filters kind of look for patterns, things like edges or curves or textures. And it also will subsample. So it will look globally. It will also, use those little sliding scale windows, slide those across the image. Here's just an example of a very simple image and a little sliding scale window, which is looking for curves. And here we see it sees a curve if we look at the array, and then that's going to be moved along the entire image. And then each of those little sections will be transformed into some numbers, those numbers into arrays, those arrays into images. And as that happens, as the images are convoluted, and then the convoluted images are convoluted, et cetera, we end up with increasingly complex and strange looking convolutional layers. So this picture of a chicken being convoluted into all sorts of crazy things. Here on this very bottom one, you can kind of see its little chicken head and beak. And then what happens is that this convolution repeats. The image is translated into many images, where each image now represents some sort of descriptor of patterns. And then those patterns are then themselves made into an array that's matched up against the original of the convolution. So I'm like probably oversimplifying a lot, but it's all comes back to kind of pattern matching. But here we've taken one image and we have sort of expanded it informationally into hundreds of images, which now have their own kind of numerical values associated with them. So hopefully that made any sort of sense. Okay, so let's go back to, we've got our segmentation and we're looking at some cells. Now, what do we do here? So we've got our initial segmentation, maybe we've got cells, we've got nuclei, and we've got cells. So we can take the nuclei, we can use those as like a seed to grow the cells, which that really helps. We can also do that with a cytoplasmic label. We could use another label, maybe label the mitochondria to define organelles, and would again do that using thresholding and masks to say, okay, you give me all of the mitochondria that are in this cell. We can define subcellular regions, only the cytoplasm, just the area by the membrane, just the area outside the nucleus. We can look at, the free space, so do what's called Voronoi tessellation, so like growing bubbles until they touch. How much free space is there? How much are the cells touching one another? Do they have neighbors? And again, remember, once we've got segmentation, now we can measure anything. We can measure the size and shape of our objects, of our regions, their orientation, their relative positions. Anything we've stained, we can measure its intensity in every region. We can also measure things like what's called the texture of the signal. So that's saying, like, what's the distribution of pixel intensities? Remember, the PCNA could be kind of uniform or it could be speckly. So is it homogeneous, heterogeneous? Are there spots? Are there stripes? And that gives us a ton of information. So what do we get out of this is very, very large text files. So what do we do with all these numbers? Well, yeah, we can do all sorts of things. Depends what you want to know. So we can ask, how do samples compare with one another? How do they compare with a whole data set? We could look at how do samples compare with known controls, and we could look at, population level. We could look at means. We could look at individual subpopulations. We can ask things like, do complex phenotypes predict things like a disease or a cancer prognosis? And then we can also look within these, sort of within the rows and columns and ask, well, are there any patterns of relationships between the things that we've measured in the data? Does having a large nucleus have anything to do with having lots of spread out mitochondria? And there could be conditionalities in there, which we wouldn't necessarily see if we looked at the means, but we could see if we look at actually the individuals. So when I say information rich, I really mean very information rich. Okay, so how do we use this big data? So one way to do it is to group things by likeness. And hierarchical clustering is a tried and trusted way of doing that. So there's various ways of doing hierarchical clustering, but these groupings will give you similarity between samples or between individuals, which are based on some patterns of values for measured parameters. In this case, they are looking at the shape of some stem cell clusters, but you will have also seen these types of clusters often for gene expression data or proteomics. So there's other ways. So we can also do what's called dimensionality reduction, right? So you've done RNA-seq on the whole genome. Every sample has, 30,000 measurements, or we've done morphometry and every cell has 200 measurements. All right, well, I don't know about you guys, but I can't visualize a graph in 200 dimensions. I would love to be able to do that, but it's beyond me. So there are things that we can do for visualization to reduce dimensionality. So one that you probably encountered is principal component analysis. And so in PCA, we're looking at all of the things that we've measured. We're trying to maximize the variance of the whole set in order to group things in terms of how far they are from some eigenvectors in this kind of imaginary space that we've created. There's also LDA, linear discriminant analysis, which is it's instead of looking at the variance of the whole set, it's trying to kind of minimize distance between groups. It's kind of like the dimensionality reduction version of Otsu's method. And these reduce dimensions and they can give you some clusters, but they don't retain the data structure. So like every time you do PCA, your PCA plot could look quite different. And something that is like the groupings aren't necessarily oriented in space the way that they would be like in a hierarchical cluster. And with PCA, so each principal component tells you how much of the variance of the whole data set is encapsulated. So usually PC1 is quite high and then it tails off. And so in a PCI plot, you can get nice clusters or you can get a big schmear, which is not very useful and doesn't look like much. But there are other ways of doing this. So those are kind of the older methods. Newer methods, we've got things like t-SNE and uniform manifold approximation and projection UMAP. So what are these newer methods? I definitely don't have time to get into the maths and I wouldn't do them justice. I'm not a mathematician. But they are nonlinear ways of reducing dimensionality and they're using machine learning in an unsupervised way. So they're very commonly used for like transcriptomics, RNA-seq, single cell transcripts. And what's really helpful about t-SNE and UMAP is that they retain some sort of the relative distances or relative groupings in terms of the similarity between groups. And so you can get sometimes a lot more informative groupings this way. So here I just want to end. How am I doing? I do want to leave time for questions, so I won't go on for too much longer, but I wanted to just give you a couple of examples of people doing this in the wild. So here's one that I thought was very interesting, which is a paper that showed that morphotypes. So the morphology of cells grown from single clones were to some extent heritable and were predictive of metastatic potential. So they took these nasty triple negative, very nasty aggressive breast cancer cells, did some serial dilution so that they got individual clones, and then they stained them to label actin and DNA, took loads and loads of pictures, did the automated segmentation, extracted all these morphological features, and then the morphology was classified from the extracted features using PCA, and also some k-means clustering in order to define what they call morphotypes. Oh, there's an and there that shouldn't be. So these different clones ended up giving rise to cells which resembled one another to a certain extent and were different from one another. So some were very small, some were big and flat, some were this very elongated spindly morphology, and each set of clones had kind of representative or characteristic morphology or kind of sets of morphology, morphotype groups within them. So then they did a couple of more experiments. So they took the different clones and they looked at gene expression profiles. So they did, I guess, RNA-seq on these different clones, and they found that the gene expression landscape, which here is expressed in the principal component space, grouped these different clones into 4 main groups, and they were different from the parental, and they corresponded to these different morphotypes, with the exception of clone number 2, which was represented across 2 different gene expression groups. And then they also took these different clones and injected them into mice and saw which ones were more or less metastatic. And what they found was that to a pretty impressive extent, both the morphology and the gene expression signatures were predictive of metastatic potential and were pretty well retained over many generations, like many passages in culture, and then also being put into a mouse. So here they were using PCA. You can also use like deep learning and t-SNE. So here there's a link at the bottom. So this is using a combination of QPath, which I talked about, and a t-SNE classifier called cytomap. So they use the QPath to identify cells and things, objects, areas in this complicated histology section with lots of different stains. Did the t-SNE to find classes. Then they converted these t-SNE classes, transformed them back into an image. And so then this image of which t-SNE class so becomes another channel. And so now you can do things like measure, okay, the expression of the CD whatever marker, and oh, it's in a certain class, and oh, it's also localized here in space. So maybe like these 2 cells are always next to each other, and there's a stromal cell that's over here that's class 1, and a stromal cell far away from the tumor that's class 3, or what have you. So again, a number can be an image can be a number, and we can transform them, and we can work with them as arrays of numbers, or we can work with them as arrays of numbers that are represented as an image, which I just think is a very fun thing to do. And then I'll just end with one which is kind of topical and uses UMAP. So this is a paper that came out about 1.5 years ago, where they used deep learning and UMAP to screen previously approved drugs to see if any of them could be repurposed against SARS-CoV-2. And so what they did was they took some cells, were either infected or not infected with the SARS. I can't remember if they used actual SARS-CoV-2. I think they did, rather than a pseudovirus. And they stained for various things. So they stained the cells, they stained the nuclei, and in purple here is a viral protein. So they were looking for treatments across this compound library where they had more or less infection. And then they did a bunch of stuff to then confirm that these drugs also worked in other models. But what I think is really cool about this is that they didn't just say, percent of dead cells or percent of infection. They used UMAP and looked at things like what were the shapes of the infected cells? What were the kind of the spatial relationships? Was it one cell infected here and one over there and one over there? Or were they always in kind of these big assemblies? Were they like dying? Were they kind of fused together? And what they found was that when they used UMAP, they could find these clusters, and those clusters actually corresponded to different ways that the virus could infect cells. So either being released through the medium, or traveling from cell to cell, or even by causing multiple cells to fuse together. So not only did they identify some drugs which were effective at preventing or limiting infection, this analysis also gave insight, which is really important, into the drug's mechanism of action, what they were actually doing to the cells. So I think this is actually just a really powerful technique if you want to, not just do a screening and kind of get yes and no answer, but actually kind of do a little bit of a deep dive into, what are we seeing? How does this actually work? So take-home messages: microscopy and any other images, they are information rich. Images are arrays of values that can be classified, and any array of values could also be an image and vice versa. And this is a really powerful tool for biology and medicine, and other kinds of science too. I don't want to leave out the chemists and the soft materials. My mind is churning away on ways that image analysis can be used to tell you things about your materials. And I know that actually this kind of, especially deep learning segmentation, is widely used for materials characterization. So like my stepdad, for example, as a material scientist, and used this type of approaches to look at particulates, and also to look at like the makeup of steel alloys. So really wide applications. Anytime you've got a picture or an array of numbers that could be a picture, these methods could be right for you. And I will finish there.